I am Bethany Young-Holt, and I direct the Initiative for MPTs. The initiative is a product-neutral, international collaboration of experts working together to advance the growing MPT field. We provide a platform aimed to raise awareness around MPTs and address technical challenges and issues facing the field. There are about 70 people registered for this webinar today, so as we allow people to join in, I'd like to spend just a few minutes providing a bit of context to the webinar. As you know, MPTs are innovative breakthrough solutions because they deliver simultaneous prevention of at least 2 health risks: HIV, other STIs, and unintended pregnancies. MPTs provide a unique and critical opportunity to leverage women's need for and willingness to use contraception with prevention of HIV and other STIs. Indeed, we're hearing from women in high HIV incidence areas around the globe that they want an all-in-one product or an MPT for family planning and HIV prevention. We're hearing women tell researchers that adherence to HIV prevention products would actually increase if it was combined with a contraceptive. There are a number of MPTs in various stages of development, with several in early-stage clinical trials. While the aim of the MPT field is to have an array of products that can address a variety of sexual reproductive health indications, the current priority indications for MPTs are HIV and contraception. Now, the MPT field has embraced novel and innovative thinking to achieve the value potential of MPTs, but there are inherent complexities with the development of such approaches. Beyond the complexities of just trying to bring together 3 distinct fields focused on contraception, STI prevention, and HIV prevention, which have historically worked relatively autonomously, there are numerous other issues that add to this challenge. These include technological challenges, such as bioequivalence and bioavailability issues that will be the focus of this webinar. There are social, behavioral, and marketing issues critical to ensure that MPTs are those that the target populations can and will use, the need for advocacy, and then also issues around product launch and introduction. So the initiative for MPTs serves as a product-neutral platform that aims to tap upon the expertise of a multidisciplinary cadre of experts to inform the clinical and social behavioral decisions and next steps for the field. As many of you are aware, several years ago, the initiative conducted a survey among MPT experts to identify the most urgent research gaps for the field worldwide. Issues around MPT clinical trial design and evaluation were identified as high priorities for the field and were the focus of a 2-day technical workshop held this past September in conjunction with a technical meeting on hormonal contraception and MPTs. So recognizing the inherent and growing complexities of developing, testing, and delivering MPTs to populations at risk, this technical workshop was designed to inform an overall strategy for the identification and successful clinical evaluation of MPTs that can reduce the risk of HIV, unintended pregnancies in high-risk adolescent girls. These efforts are especially urgent in the context of the evolving HIV prevention field. So through presentations and lively discussions, the diverse experts at the workshop reviewed and debated a range of issues critical to addressing the potential market impact and clinical evaluation of MPT products. This report is available on the IMPT website. So the importance and complexities around bioavailability and bioequivalence strategies for MPTs that were generated a great deal of interest and discussion during the September clinical trial design workshop. Today's webinar builds on those discussions at the workshop and focuses specifically on critical conversations around bioavailability and bioequivalence strategies, including feasibility and risk. Joining us today is Charles DiLiberti of Montclair Bioequivalence Services, who presented on bioequivalence during the September workshop, and he will be giving the main presentation. today. In addition, I'm delighted to welcome Dr. Laneta Dorflinger and Dr. Joe Romano, who will be leading a facilitated discussion. Dr. Dorflinger is the director of FHI 360's Contraceptive Technology Innovation Program, and Dr. Romano is senior scientific advisor to the Initiative for MPTs and is president of the NWJ Group. I am now pleased to introduce Charles DiLiberti, president of Montclair Bioequivalence Services. Mr. DiLiberti has over 30 years' experience in the pharmaceutical industry, the majority of which were at Barr Laboratories. Mr. DiLiberti built and oversaw Barr's cutting-edge bioequivalence and pharmacokinetics program for generic drugs, small molecule proprietary drugs, and biologics. In 2010, Mr. DiLiberti started his own firm, Montclair Bioequivalence Services, which provides strategic consulting services around the world in generic, innovative, and biologic drug development, biostudy planning, risk management, formulation, development guidance, and advanced troubleshooting methods. With a focus on complex and difficult issues in women's healthcare products. He holds a BA in Biochemical Sciences from Princeton and an MS in Chemistry from Stevens Institute of Technology. So Charles, we're honored to have you, and thank you.
Thank you very much for that glowing introduction. If we could please advance to slide number 12, the disclaimer. I'll give you a moment to read this. The main thrust of this is that development of any drug, in particular these MPTs, is a very, very complex operation, and it's certainly advisable to seek expert advice before embarking on any critical decisions. Today is just a high-level overview. We'll be scratching the surface of bioavailability and bioequivalence principles. Please advance to the next slide, which should be number 13. So the general concepts of bioavailability and bioequivalence hinge on the idea of how much drug is getting to the site of action. For bioavailability, it's literally the amount of drug that gets to the site of action. You could think of this as, for example, 50% of the drug that's administered gets to the site of action, 70%, 100%. Bioequivalence is a little different concept. It's a comparative concept where you would have a reference product that has an established safety and efficacy history, and you have a test product that's under development that you want to link to that reference product. And the basic concept of bioequivalence is to demonstrate that the amount of drug that gets to the target tissue for your test product is comparable and close enough to the amount of drug that gets to the target tissue for the reference product, so that you can then infer that the two products will have comparable clinical effects. Bioequivalence is sometimes referred to as a bridging type of study. The next bullet underscores that it is a useful tool to help to reduce the clinical study burden, but please, it is not a panacea. And I hope to dispel some of those perhaps overly optimistic views of bioequivalence today. In addition to the amount of drug that ends up at the target tissue, the other piece of this is that the rate at which that drug appears at the target tissue is important, the rate of drug delivery. Just one general concept before we move on is most of my talk is focused on U.S. FDA matters and regulations. And although other regulatory agencies around the world might have some similarities in their views, each regulatory agency has its own unique perspective sometimes. Let's proceed to the next slide, which should be number 14. In general, drugs and drug products can be divided up into two broad categories, those that act systemically and those that act locally. This slide, the upper half, illustrates systemically acting drug products. where you apply the drug to a particular part of the body. Often these are ingested orally. The drug is then absorbed, goes through the bloodstream to the site of action. Now, in terms of bioequivalence, as per the previous definitions, obviously we would like to be able to monitor the drug concentrations at the site of action, but it's rather impractical to ask people involved in a study to donate a piece of their brain tissue for analysis. So in lieu of that, for systemically acting drug products, we would typically collect a series of blood samples in the time course following dosing and measure drug concentrations in those blood samples. This is relatively easy to do, and because the bloodstream in this type of drug product is upstream or before the site of action, the blood concentrations are typically a pretty good proxy for the target concentrations in the target tissue. Next slide, please. So in contrast, a systemically acting drug, and now we're on the lower half of the slide, locally acting drug on the lower half of the slide, you apply the drug at a particular point of the body, and let's use, for example, a topical cream that's used to treat a skin condition, such as acne or rosacea. So you apply the drug, and it goes basically directly to the site of action. And it may or may not ultimately appear into the bloodstream, which is why I have a dotted line going to the bloodstream box. Just as with systemically acting drugs, we do want locally acting drugs to work at the site of action, so we want to be able to monitor drug concentrations in the target tissue. But this, like systemically acting drugs, is hard to do. In contrast to the systemically acting drug situation, we have some hesitance to rely on blood concentrations, which may or may not even be detectable, to assess drug concentrations at the site of action, simply because the bloodstream in this case is downstream from the site of action. I've underscored the word product in both of these situations just to make it clear that it's not so much a matter of the drug substance that is administered. You can administer the same drug substance via two different routes, and in one case it may be a systemically acting drug product, and in the other case a locally acting drug product. Next slide, please. So we should be on slide 16. I'm going to cover a few slides now on the systemically acting drug product situation. So when we measure pharmacokinetics, or the movement of drug through the body, we typically collect a series of blood samples over the course of time following drug administration, and that enables us to plot a graph of drug concentration in blood on the vertical axis versus time. And typically, immediately after the drug is administered, it rises rapidly to a peak and then falls off, usually somewhat more slowly, often in exponential fashion. And there are two key features that we look for in pharmacokinetic or PK studies. One is the maximum drug concentration, or Cmax, and the other is area under the curve, which is literally the area under this plasma concentration versus time curve. Now, Cmax is used as a surrogate or proxy for the rate of drug administration, considering that typically when you have a drug that's absorbed more quickly, it yields a higher peak, and conversely, a more slowly absorbed drug gives a lower peak. Area under the curve is used as a proxy for the extent or total amount of drug absorption. Next slide, please. Should be on slide 17 now. Now, for bioequivalence for systemically acting drug products, again, we're trying to link clinical effects of one product under development to those of a reference product, and typically we do this by assessing the ratio of Cmax of the test product to the Cmax of the reference product. Similarly, we measure the AUC of the test product divided by the area under the curve of the reference product. So in almost all cases, we'll be looking at test to reference ratios. It's generally considered that about a 20% difference in pharmacokinetic response for Cmax and AUC is typically seen to be clinically insignificant. So as long as we're within that band, it's not a big issue. We want to control the product so that we don't fall outside of that band. So this corresponds to a test-to-reference ratio of 80 to 125%, and you might note that that's an unequal interval on the two sides of 100%, and that's simply because 125% is the reciprocal of 80%. So this can also be viewed as the test-to-reference ratio is no lower than 80%, and the reference-to-test ratio is no lower than 80% either. Next slide, please. Slide 18. So one of the hardest things to appreciate in bioequivalence is the concept of population statistics versus sample statistics. The overall goal of a bioequivalence study is to measure the pharmacokinetic response in a small group of volunteers or patients, and from those results to draw conclusions about the overall population. So we're looking to draw conclusions about the population average of the test product, for example, for Cmax, and the population average of the reference product. But these population means are unknown and unknowable because we would literally have to test every human being on the face of the earth in an enormous study to determine these means. So what we do instead is to estimate from our small sample what those population means are, and we can put error bars around those, or confidence intervals. And this is much like what's done with public opinion polls, where a small subset of, for example, the U.S. population is polled, and they extrapolate results to the overall U.S. population with error bars. They typically, plus or minus a certain percentage margin of error. Very similar situation for bioequivalence. Next slide, please. Should be slide 19. So the criteria that are typically applied are that the 90% confidence interval for this test-to-reference ratio has to fall entirely between 80 and 125%. Now, to help wrap our heads around this, the consequence of this and the intent of this is to make sure that there's less than or equal to a 5% risk that the population test-to-reference ratio is truly less than 80%. And similarly, there's no more than a 5% risk that the population test-to-reference ratio is greater than 125%. And this is typically applied to Cmax, area under the curve, and sometimes to other pharmacokinetic metrics. Now, what this means is that we have to have a high degree of confidence going into a bioequivalence study that the true population test-to-reference ratio is really within about 5% of perfect, of 100%, in order to have a reasonable chance of passing with a reasonable number of subjects. There's a lot of misunderstanding in what these criteria mean. Many people think that the bioequivalence criteria mean that it's likely that the test and reference product truly differ by 20 to 25%. However, this is not the case. These criteria actually make it extremely unlikely that the test and reference product are truly that different. And we'll see that in a moment. So passing these bioequivalence criteria is actually very rigorous and difficult unless the two products are engineered basically to be almost identical. Next slide, please. This should be slide 20. So if we look at a plot of what's called the power or probability that a bioequivalence study will pass, which is on the vertical axis, and plot that against the true population test-to-reference ratio for a parameter, let's say Cmax, that's on the horizontal axis, we get a bell-shaped curve. That's the blue bell-shaped curve. And although we never know the true population test-reference ratio, we can mentally think if we were to test the reference product against itself, we would know that the true test-to-reference ratio was 100% because it's the reference compared to itself. And in that case, the bioequivalence study would have a very high chance of passing, let's say almost 90%. So in real life, we don't know what this value is, but we, based on in vitro testing and formulation design, we tend to assume that the test formulation will be within, say, plus or minus 5% or plus or minus 10% of the reference product performance. And so that's represented by the green band. And as long as we're within that region, we have a reasonably high chance, maybe about 80% chance, that this bioequivalence study will pass. However, if we're wrong, and if the true test to reference ratio is lower, is further off than 100%, then the chances of passing this bio study fall off precipitously. And it shouldn't be surprising. This bioequivalence criterion is a gatekeeper to ensure that we really reject poor quality products that differ by 20% or more. And you can see if the true population varies or differs from the test product differs from the reference product by 20%, like the 80% point, the chance of passing this bio study falls down to below 5%. So this is a very stringent tool and very stringent criterion to meet. Next slide, please. So I've just outlined what happens with systemically acting drugs. Now I'd like to turn to locally acting drugs. So with a locally acting drug, again, you apply the drug. It basically goes directly to the site of action and may or may not end up in the bloodstream. Just as with systemically acting drugs, we're still under the mandate to assess drug concentrations at the site of action. But although it might be a little bit easier to collect a piece of skin than a piece of brain, it's still hard and largely impractical. So we typically don't do that. However, unlike the systemically acting drug situation, with locally acting drugs, the drug concentrations in the bloodstream may not be such a great proxy for the concentrations at the site of action. Next slide. Slide 22. So what do? We generally can't use drug concentrations in the blood. So then we're forced to rely on clinical effect or some other surrogate marker of clinical effect. Now, this often has very poor sensitivity towards formulation differences, as I'll show in a moment. Although bioequivalence studies on systemically acting drugs are often, but not always, done on normal healthy volunteers, because you're looking for a clinical response for a locally acting drug, you will almost always need to use patients. So that makes it somewhat more difficult. In addition to the difficulty of ethical considerations regarding collecting bits of tissue from volunteers or patients, there are other technical considerations that make it highly impractical to measure drug concentrations in tissue. And one of these is that the drug is applied almost in direct contact with the site of action, so you have a huge risk of contaminating your tissue sample with drug. Next slide, please, number 23. I'm sure many of you have seen a dose-response curve. This is a typical sigmoidal-shaped curve which plots clinical response on the vertical axis versus the logarithm of the dose on the horizontal axis. Ideally, when we're measuring bioequivalence, we would want to do it in patients. This is, again, for locally acting drugs. We'd want to do this in patients at a point where the dose-response curve is relatively steep, so that small changes in dose or availability at the site of action result in a relatively large clinical response that we can then measure. However, this is not a good point on the dose-response curve to be for a drug used to treat patients, because you don't want typically a drug to be very sensitive to how a patient uses it or what their body mass is or whether they're male or female or any such matters. You want the drug to work and work reliably, which means that typically for many marketed products and approved products, the dose-response curve, we're dosing patients at the upper end of the dose-response curve. Great for patients, but very a very poor place to assess bioequivalence, because then it takes a very, very large difference in drug availability at the site of action to elicit a relatively small clinical response difference. Next slide, please, slide 24. So for locally acting drugs, we covered systemically acting drugs and the typical requirements. So for locally acting drugs, these criteria that I'm about to mention are those that are defined for bioequivalence for generic drugs. And the reason I'm focusing on this is because they have been much more standardized and are more consistent across different products than bioequivalence criteria for locally acting drugs in a new drug setting. There's more variability in practice, I would say, in the new drug setting. Similar principles are used, but the actual implementation might be a little different. So anyway, for a clinical endpoint bioequivalence study, again, for a locally acting drug, it has typically 3 criteria. So you give a test product, you give your reference product, and you give the placebo. And you have to show statistical equivalence of the test and reference products, statistical superiority of the test product to placebo, and superiority of the reference product to placebo. The particular endpoints used and acceptance criteria may vary by product, but these are sort of the basics. Now, this threefold criterion can actually be more challenging than the criteria for the original approval of the reference product itself. Typically, when you get the reference product approved, it's just a matter of showing superiority of the test product to placebo. You don't have these additional criteria of, reference to placebo and test and reference equivalence. So this is actually a pretty hard criterion to meet. Now, in addition to showing clinical effect, which is sort of a measure of efficacy, often if locally acting drug products are also absorbed into the systemic circulation, even though they don't act that way, you also would typically have to show some sort of bioequivalence criterion on the systemic blood levels for Cmax and AUC, more as a measure of comparable safety rather than efficacy. Next slide, please, slide 25. Not that necessarily all MPTs are going to be vaginally administered, but quite a few are, so I wanted to just touch upon some of the peculiarities of vaginal drug administration. So vaginally administered drugs can fall into various dosage forms. You can have tablets, suppositories, inserts, creams, gels, rings, etc. So the big question is, are these systemically acting or locally acting? Next slide, please. We should be on slide 26 now. The answer is, it depends. Now, based on the numerous FDA bioequivalence guidances that have been crafted for generic drugs, if you look through those that are administered vaginally, almost all of them, the vaginal administration is treated as a locally acting drug. For example, drugs targeting the vaginal epithelium or the inside of the vaginal cavity, the lumen, for example, to treat vaginal atrophy, dyspareunia, bacterial vaginosis, etc. These are obviously going to be locally acting drugs. They act at the site of administration. Drugs associated with pregnancy other than contraception are typically also treated as locally acting drugs, even though it might be less clear why. Now, the exception here is that vaginally administered hormonal contraception seems to have been considered to be largely a systemically acting situation by FDA. Next slide, number 27, please. So, in my view, MPTs are probably going to be likely to be considered to be both, in some respects systemically acting and in some respects locally acting, with the hormonal contraceptive component, assuming they contain one, probably being deemed to be systemically acting, and if an anti-HIV component will probably be deemed to be more locally acting, though not necessarily so. Again, very highly product dependent. Next slide, number 28, please. So in terms of how we would demonstrate bioequivalence for MPTs and perhaps reduce the clinical burden somewhat, I think you might possibly consider a conventional bioequivalence approach for the contraceptive component, but there are many, many constraints about this. You'd have to have the same route of administration for the test and reference product. The plasma concentrations of the test and reference product would have to be essentially superimposable. This is going to be a very difficult standard to meet. Possibly an alternative approach might be to bracket. And by bracket, what I mean is that the entire PK profile of the test product should ideally fall between the PK curves of two approved reference products. In other words, one with a higher plasma concentration and one with a lower plasma concentration. This might essentially allow you to widen the goalposts. Ordinarily, the goalposts, when you compare against a single comparator product, are pretty narrow. But if you can show that you sort of thread the needle between two different approved products, you might be able to make it a little easier on yourself. However, this is something that would certainly require negotiation with FDA or the other regulatory agencies. And, of course, you have to have the two comparator products available and with proven safety and efficacy and appropriate blood levels, et cetera. Next slide, please, number 29. Vaginal drug administration has its own unique PK issues that I just want to touch upon briefly. Typically, drug absorption is somewhat slower than oral absorption, whereas when you give a drug orally, many drugs are metabolized quite a bit before they ever hit the systemic circulation. They're metabolized by enzymes in the intestinal wall or in the liver. Obviously, vaginal absorption significantly bypasses this first pass effect. Vaginal rings, in particular, tend to have what's called the burst effect, where when you first put them in, there's a huge release of drug, which then tapers off over the course of time. We have to consider the state of the vaginal epithelium because that is a limiting factor in drug absorption vaginally. So a normal, healthy, premenopausal vaginal epithelium is a pretty good barrier to absorption of drugs and tends to limit how much drug gets in, whereas a postmenopausal vaginal epithelium tends to be much more leaky and allows much more drug in. And when you have women in the perimenopausal state, then it's unclear as to how much drug is going to get in. Obviously, certain types of dosage forms like creams, gels, suppositories, there's a risk of, this is a very real factor, the physical loss of a portion of the drug. Depending on how much you put in, it can actually leak out. We also need to consider interaction with concomitantly administered vaginal products and OTC products. For example, if a woman is on an MPT and she contracts a yeast infection, needs to be treated with the vaginal medication for that. How does that interact with the MPT? And finally, there are some cases where FDA has required drug transfer studies where the concern is exposure of the male partner during intercourse to the drug that's administered to the female. Next slide, please. Slide 30. We're coming down the home stretch here, and I don't want to belabor this point, but we have to be careful in devising a dosage form of the inactive ingredients. It's much easier for FDA to accept the safety of a product if the inactive ingredients or excipients have already been used via that route of administration and in the quantities that are proposed. If you start to introduce novel excipients that have never been used or never been administered via the proposed route of administration, or if you use existing excipients that are used in higher concentrations, that raises red flags at FDA and it might prompt the need for a safety study or an animal study. So it's best to stay within what's called the inactive ingredient database limits. Next slide, please, slide 31. So Turning now to what I would expect the bioequivalence criteria for the locally acting or antiretroviral component of an MPT would be, I think this is going to be a difficult proposition. I understand that there might be some products where the antiretroviral might possibly be thought of as being systemically active. But again, this is going to depend on negotiation with the FDA or regulatory agencies and the particulars of the drug itself. Bioequivalence is more than just a study. It's the concept based on totality of evidence. So we need to factor in not only just the clinical endpoint bioequivalence study if that's needed, but also what's the dosage form, what are the active ingredients, is the composition same or similar to the reference product, does it release drug in vitro at the same rate as the reference product, does it produce comparable blood levels as the reference product. And also a clinical endpoint bioequivalence study can sometimes use an easier marker of efficacy than, for example, might be needed for a full-blown phase 3 type of study. You know, this is not directly relevant to the case of MPTs, but you can envision that if you're giving a medication to reduce blood pressure, it's a lot easier to measure blood pressure response than it is to measure the effect on mortality. So a similar concept might be applied to a bioequivalence study for a locally acting drug component. This is all going to require extensive negotiations with the regulatory agency. Although there might be some possibility of some relief of clinical burden by in vitro and PK-based bioequivalence approaches, I still think there's going to be a high hurdle to overcome here. Even if you can demonstrate bioequivalence, the regulatory agency is still likely going to want to look at some sort of safety data with your overall product as intended. Next slide, please. So this should be the last slide in my deck. Bioavailability and bioequivalence aspects of the probable clinical program for an MPT. This is going to be highly drug and dosage form dependent. We've talked about bioequivalence potentials for the contraceptive component. It might possibly work. Again, very product and dosage form dependent. It depends on what reference products you're trying to link to. Bioequivalence for the locally acting component, assuming the anti-HIV component would be one, it's going to be more challenging. Now, the use of bioequivalence approaches to reduce a clinical burden, again, it's probably not going to be a panacea. It may be more of an incremental benefit. It may reduce the clinical burden rather than eliminate it. Trying to show primary safety and efficacy for an MPT using this approach is going to be rather difficult. However, there is a more likely application in a secondary bridging kind of scenario. For example, to extend the primary phase 3 conclusions of safety and efficacy to subpopulations, for example, pediatric, perimenopausal, renally impaired, etc. This is a more likely scenario where a bioequivalence approach would be more acceptable to FDA. And even regardless of how you show bioequivalence, assuming it's via a clinical endpoint, FDA typically still also wants you to evaluate the systemic drug availability of the proposed product just as a measure of safety and also just to characterize it. So with that, I'll conclude my presentation. Next slide is thank you to all for your patience and attention, and I'll turn it over to the organizers.
Thank you for presenting on this topic, Charlie. We will now begin the facilitated discussion portion of our webinar, which will be led by Laneta Dorflinger and Joe Romano. Laneta and Joe, would you like to begin with a few questions or a brief reflection of the presentation?
Yeah, hi, this is Joe. Thanks, Charlie, for that presentation. I do have several questions, and I don't want to monopolize the discussion. Laneta, did you want to kick off with some thoughts? I know we had some earlier discussions. Well, perhaps Laneta is muted. I am happy to kick off with a couple questions, somewhat technical in nature. Charlie, you mentioned that the primary comparator metrics between the test and the reference products are the Cmax values and the area under the curve. What does that mean in terms of the feasibility of using a bioequivalence approach when trying to compare products that are perhaps reformulations where the reference product has a shorter duration of effect than the new test products? You have something that's, X hours or X days, and that's approved, and that's your reference, and you've tried to improve the formulation so it can now last many more hours or many more days or weeks. How do you superimpose those AUCs for comparison's sake, or is that just a bit of a bridge too far for a bioequivalence strategy?
Thank you, Joe. Excellent question. The short answer is that's trying to bridge too far. When you intentionally change the release characteristics of a product that changes duration of action, already you're in territory where you're going to have it's going to be essentially impossible to match on the peak drug concentration. What has been done, for example, in orally administered products is there have been studies, for example, to link repeated, more frequent administration of an immediate release product to a less frequent administration of a modified or controlled release product. Although, I think that's going to be even more challenging in this area just because of the limited availability of reference products. So a short answer is I think you're outlining a very challenging situation. It's much more likely that you'd get more traction with bioequivalence where the test and reference product are designed to have the same profile.
Okay, thanks.
Thanks. Charlie, can you hear me?
Yes.
I think, okay, great. I was somehow on mute before and wasn't able to get in, and I want to thank you for such a great presentation. One of the questions I had as I was listening to you is how your presentation makes us think so carefully about how these BE or BA approaches could be applied, and particularly the bracketing approach potentially to reduce the burden of full efficacy data, and you spoke a little bit to this. I'm wondering if you could tell us whether a bracketing study could be done with different dosage forms. And I'm thinking, for example, of levonorgestrel, which is being evaluated in some of the MPT products for vaginal delivery. Could that be considered in a bracketing study versus an oral and perhaps an implant, thinking of high and low doses?
Yeah, great question. I'm not sure I have a real answer to that. I think that would be the subject of negotiation with FDA, and it would also depend on what the particular blood levels are of the test product as well as the potential bracketing comparators. But I would be a little cautious to assume that approach would definitely work, and instead seek FDA's input on that idea.
Okay. Great, Joe. Do you want to? I know you have another question.
I do. We talk about these clinical assessments for bioequivalence, and I'm wondering, and we think of them as perhaps simplifying it, but as a shortcut. It wouldn't necessarily be a trial of the size and magnitude necessary to demonstrate or redemonstrate efficacy for a prevention product. But I'm interested, what is a typical design for these things, and how big are they? Are these, for example, are they typically crossover designs so that you're comparing the reference and the test product in the same person, so that you're presumably dealing with the same metabolism and so on, and that may be a little more useful than running with a reference population or a reference arm and a test arm. If it is crossover, how many people does it typically take, and how long do they need to be on reference and test? Just wondering what we're talking about here in terms of magnitude. Are we potentially underthinking how big and how complicated these studies might need to be?
This is largely on the drug product that we're talking about and how long the duration of action is. Ideally, as you sort of alluded to, you would try to do a bioequivalent study as a crossover design if you can, because by, in essence, you're comparing then the test and reference product within the same individual, and that it can be a huge benefit in terms of reducing variability because it's a within-subject comparison. So, you know, a drug product that acts over a fairly short duration would often be compared in a single-dose study. So you give one dose of the test product, you follow the blood levels, you have a washout period, and then the patient comes back and you give a dose of the reference product and follow the blood levels thereafter. And, you know, typically half of the volunteers or patients would get the test product first and then the reference, and then the other half would get the reference product first and then the test. So it's sort of balanced with respect to order. So that crossover design is ideal. If you can do that, sometimes you can dramatically reduce the number of subjects that you need for the study because you control the variability. The number of subjects that you need for such a crossover design study can vary tremendously by product. Some products are very low variability and typically require only small numbers of subjects. For example, levonorgestrel, certainly as an oral tablet product, variability is typically less than 20%. So you may be able to design a study with only, say, fewer than 30 volunteers as a crossover design. There are other products, even though they may be short-acting and suitable for a crossover design, that have extraordinarily high variability. Not that it would be used as an MPT, but progesterone is one of the poster childs for high variability. It's a female healthcare product, and oral capsules typically have variability on the order of 80 to 100%, and you may need hundreds of subjects in the study. So it varies enormously, but almost always you're much better off with a crossover design if you can do it, as opposed to a parallel design where you divide up your group of volunteers or patients into two groups. One group gets the test product, the other group gets the reference product. So then it becomes a between subject or between patient comparison, and then the inherent metabolic variability and other variabilities between the patients can wreak havoc on your study design. And you may very well need, again, very highly product dependent, but it's easy to get up over 100 or maybe almost 200 subjects needed for a parallel design.
So that's actually encouraging. Sorry. And just, sorry, let me interrupt, just a quick follow-up. If indeed we're talking about a single dose, then the equivalence need not necessarily be demonstrated at steady state levels?
Yes, FDA typically considers a single-dose study to be a more sensitive measure of bioequivalence and drug performance than a multiple-dose steady-state study. So that's usually their first preference. Now, that said, when you're developing a new drug, it's entirely possible that FDA may want you to do both a single-dose study and a multiple-dose steady-state study.
Okay, thanks.
But that said, typically when you do a multiple-dose steady-state study, where, for example, you would give multiple sequential doses of the test product until you have achieved steady-state blood levels, and then you measure them over the course of a dosing interval, and then you wash the patient out and then switch them over to the reference product and do the same thing. You build them up to steady state with multiple repeated doses. You measure over the course of one dosing interval, and you compare the test and reference at steady state. That type of design is it typically has considerably lower variability than the single-dose design. So although it's sort of more involved in terms of how many doses you give, you get typically smaller subject numbers.
Thanks. Charlie, related, I'd like to explore a little bit the metrics. So you talked a lot about the Cmax and the area under the curve, but for some of the products that, for example, injectable products, they're injectable contraceptives, but they're also going to be injectable products for HIV prevention and MPTs potentially. So in those cases, we're really looking at, not in a therapeutic context, but in a prevention context, the importance of C-min or C time X, whatever the duration is going to be, more so than the Cmax. So Cmax may be a safety indicator, but less in the way that we think of the effectiveness indicator. Have you seen situations where you've used, let's say, a C-min value where Cmax may not necessarily meet your target VE, but your area under the curve and C-min could be more important?
Well, in the context of a new drug, you typically have a little bit more wiggle room with FDA than you do in the context of a generic drug. So there are many cases throughout history where new drugs have not quite met strict bioequivalence criteria. In one case might be what you mentioned, where you don't quite meet it on Cmax, so some other argument is put forth to sort of cover that deficiency. So yes, I mean, what you're describing is entirely a plausible argument. Will it necessarily succeed? Again, it's going to depend on the particular drug and the scenario. But obviously, if you have a drug that's dosed chronically and you're concerned about steady state levels, then yes, C-min is an important driver of efficacy. So that said, you can often infer that if you meet Cmax and AUC under single-dose conditions, you will sort of automatically be okay on Cmax and C-min and degree of fluctuation and all the other parameters at steady state.
Thank you. Are there any questions from the audience? We have a few more minutes. We've not had anything come in.
I have one more, if I'm not being too greedy with regard to time, but this is a very important subject, particularly for the MPT field. You gave some examples, Charlie, of the kinds of comparisons that are typically done during the course of your presentation, and that included both systemic as well as locally active drugs. The takeaway message I got was that there were real limitations on how different things can be before you're beyond the limits of what's going to be acceptable for a bioequivalence demonstration of equivalence. In this field, we're talking about existing contraceptive products and existing HIV prevention products, and we have an oral HIV prevention product that's using tenofovir, and we have a vaginal ring that's using dapivirine that hopefully we have approved very soon. And then we've got one vaginal ring with two hormones in it, and then implants and oral pills, and et cetera, with different hormonal contraceptives in it. I'm guessing that our bioequivalence options, particularly on the HIV side, are very, very limited in the absence of approved references of similar dosage forms to what's being contemplated for MPTs. Is that a fair assessment of our status?
Yeah, I think so. Again, I think that probably the action of the vaginally administered antiretrovirals is going to be viewed largely as a locally acting phenomenon. So that automatically makes things harder. The number of comparator products with the same active ingredient is much more limited. So yes, I think the HIV component is going to be the more challenging of the two.
Should the piperine ring be approved, that at least provides a reference formulation for future piperine products.
Yes.
But we don't really have that on the tenofovir side at all.
Yeah. These are going to be challenging. And, again, I think we need to think of bioavailability and bioequivalence in terms of reducing the clinical burden and, just sort of conceptually thinking, okay, if you could not link to some approved product via, close bioavailability or bioequivalence, if you could not do that, what would your clinical program look like? Then you're talking about basically proving safety and efficacy from scratch. Whereas you might get somewhat of a break on the scope and size of your phase 3 studies if you can do some sort of linkage, whether it's a perfect linkage with strict bioequivalence or even getting close, showing close relative bioavailability. I think it's going to be a help.
Right. Okay, one last chance for anybody in the audience to offer a question. It seems like there was a question or two that I covered by chance. But in the absence of that, I think we could conclude. Or Laneta, anything from your side?
No, but I just want to thank you again, Charlie, because I think you've helped define the challenges that we have certainly in this field, and I appreciate the clarity of your presentation and your thoughts.
Well, thank you so much to Charlie for his excellent presentation and to Laneta and Joe for facilitating today's discussion. In addition, thank you to all of today's participants for your thoughtful questions and all that you do to advance the lives of women and girls around the world. A recording from today's webinar will be available on the IMPT website shortly. Thank you.
Thank you all.
Thank you.